Okay, well, good morning everyone. Nice to see you today. We now come to the last couple of lectures in this block of content on oxidative stress, and I guess because you're involved in a pharmacology course, you're probably interested in knowing whether we can do anything to block some of these oxidative processes that contribute to DNA and protein and lipid damage in cells. So that's what I want to talk about in these couple of lectures. And of course you're also addressing this sort of basic hypothesis in your lab classes in your drug discovery project. So hopefully what we're talking about today and tomorrow, or at least Thursday, will help you with your research projects, knowing what the goalposts are. Okay, so I've decided to cover this content firstly in this lecture on antioxidants, and then on Thursday we'll look at carbonyl trappers. Now of course antioxidants is just a term itself, is one that lots of people use, and it's pretty commonly exploited in marketing of both pharmaceutical products and alternative healthcare products, and even foodstuffs sometimes come with labels claiming to have antioxidant properties. So see here, green tea, you often find the word antioxidant on the box of green tea. So what do we mean when we use this word, and at least to a scientist, what do we mean by it? Well, it's a substance, and this is from Hallowell and Guttridge in their sort of standard handbook of oxidative stress research. An antioxidant is any substance that, when present at low concentrations compared with those of an oxidizable substrate, significantly delays or prevents oxidation of that substrate. So that's a nice simple definition of a molecule that almost acts sacrificially to undergo oxidation and protect a critical cellular component. So it's almost like a Secret Service type person looking after the US president, willing to take a bullet to protect the chief. This is a basic idea that an antioxidant sacrificially reacts with a damaging oxidant and protects something more vulnerable. Now the problem with this area, and one of the reasons why we should be a little sceptical about some of these claims on healthcare products, is that it's relatively easy to meet these criteria in vitro, but then talking about an antioxidant acting as an antioxidant in vivo in the whole human body or a whole animal is more challenging. And this is going to be one of the issues you're going to have to grapple with during your drug discovery project, because all our experiments are conducted in vitro. And hopefully this lecture will set the scene for some of the issues that have to be considered when trying to claim that a molecule actually acts as an antioxidant in a human or an animal. Okay, so how do we actually go about proving then that a compound is actually an antioxidant, even just in an in vitro system? How do we go about doing that? Well, there are 4 criteria that Hollowell and Guttridge propose. And one of the central criteria is that the antioxidant needs to actually be shown to undergo a chemical alteration to its structure as a result of reacting with the free radical. So it's got to undergo you've got to be able to measure the product of a reaction with a free radical and say, yes, that antioxidant has truly been altered in its chemistry. But then you also need to show that that compound does actually act sacrificially. It does block the oxidative damage to a biomolecule. And one of the ways it's been done in the past, we didn't use this method in our labs, it's a little bit involved, but to show that a molecule protects lipids against oxidation, people have often made use of just an old-fashioned oxygen electrode, and these were sort of developed by biochemists to look at things like mitochondrial oxidative phosphorylation in vitro. And you can simply put say a mitochondria or a biological target substrate, in this case a polyunsaturated fatty acid, put it in a buffer in an airtight chamber, and then add something to stimulate the consumption of oxygen. And so in the case of lipid peroxidation, remember that after initiation of lipid peroxidation you have these carbon-centered radicals that react very quickly with oxygen. So they trap oxygen, pull it out of the solution. So once you add your oxygen or initiation system, iron and hydrogen peroxide just shown here, we start getting a loss of oxygen in the buffer system inside the chamber. And then you need to show that adding your antioxidant slows down the rate of oxidative damage to the polyunsaturated fatty acid and slows down the rate of oxygen consumption. So that's one of the ways of proving that your molecule really is an antioxidant, that it acts sacrificially and spares the lipid. But then you need to do something a bit even more challenging. You need to show the same thing happens when cells are undergoing oxidative stress, cultured cells, and also that chemistry that you identified in aim 1 up here. Can you actually show that those oxidized derivatives of the antioxidant are actually forming in a cellular biological system? And then probably the toughest criteria down here is, can you show that the administration of your antioxidant actually suppresses oxidative damage in vivo? Now another thing enters the scene that we need to talk about too, because there are now different types of antioxidants that are recognised, and in our lab class we're sort of focusing on direct acting antioxidants. These are the ones that have had most attention over the years. But we now know that there's another very effective class of indirect antioxidants, and these act at the transcriptional level to boost the expression of antioxidant proteins within cells. And so these are called NRF2 boosters because they interact with NRF2, an important transcription factor that regulates the antioxidant response genetic pathway. Okay, but first, before we come to those, we're going to talk a bit about the differences between these two antioxidant modes of action. But bear in mind, most of the literature historically has focused on direct-acting antioxidants, and there's a whole swag of them, literally. We can consider small molecules, but also high mass proteins. We don't have any time at all to go into those at all, but we could look at things like superoxide dismutase and catalase and peroxyredoxins and other important proteins that also directly inactivate reactive oxygen species. And then if we just focus on the low mass substances, we can discriminate between synthetic and naturally occurring molecules, and both those classes can be further subclassified into those synthetic agents that are created with medicinal intentions. That's sort of what we're doing in our lab class at the moment. But then there are also synthetics that are used as food additives, and then in terms of naturally occurring ones, we can consider both exogenous ones we accumulate via the diet, as well as endogenous molecules that likely act as free radical trappers. High mass proteins also have a degree of complexity. We can consider some that mainly work by trapping metals to scavenge things like iron to prevent Fenton chemistry. And then there are other antioxidant proteins that directly trap superoxide and hydrogen peroxide. Okay, so there's an awful lot of complexity in the antioxidant field. We're just sort of skimming across the surface in this lecture. Now, like I said, the classification of whether a molecule acts as an antioxidant via direct or indirect modes of action is very important these days. And some actually have both effects, and so that makes interpreting their effects even more complicated. But the idea with a direct-acting antioxidant, let's say we'll use vitamin E here, alpha-tocopherol as an example, is that it directly inactivates either a peroxyl radical or a superoxide radical. So it targets the chemical species. And it also undergoes chemical alteration to form some of those oxidised products that you can measure in vivo or in culture media. On the other hand, an indirect antioxidant, and now there's a number of these available if you go and look in the health food store or your local pharmacy, you'll find a range of products such as NRF2 Renew here, and these are NRF2 boosters that act via that NRF2 transcription factor to activate expression of cytoprotective genes, and we'll look at some examples of that mode of action in a minute. Okay, so just to clarify then the difference between a direct and an indirect mode of action. A direct acting antioxidant directly neutralises some of those nasty chemically reactive substances that are formed during oxidative stress and often are released from endogenous sources, such as activated immune cells or the mitochondrial respiratory chain. But then indirect antioxidants have a nuclear mode of action in that they, via the transcription factor NRF2, they boost this transcriptional response and boost the intracellular activity and availability of antioxidant proteins. Okay, so, and like I said, some molecules, just to make life complicated, act via both modes of action, just to complicate it. Now for the next few moments, let's just spend a bit of time looking at some of the major classes of direct-acting antioxidants, and we'll look at, and some of this is relevant, of course, to our drug discovery projects at the moment, because we're assuming the molecules act via this mode of action. Now, there's a lot of interest in these sorts of products from consumers. Everyone would like to hope, wouldn't we, that we could simply take a product that can allow us to suppress the ageing process and the role of oxidative damage in it. And it's very easy to appeal to people's vanity, and they'll fork out the money to, if they're buying a product they think can slow it all down and retain their youthfulness into old age. But of course, whenever you get money involved with pharmaceutical products, then human corruption shows itself, and we see exaggerated claims being made, not necessarily a good evidence base to support those claims. And also governments are sort of reluctant to get too involved in regulating some of these products. And so we have a complex environment where these sorts of antioxidants are freely available, but on the basis of questionable scientific evidence. And also one of the problems is that these sorts of products often get exempted from the more rigorous sort of clinical pharmacology evaluation we'd apply to other pharmaceuticals. So if you go and look at the clinical trials for some of these products, they often don't really define things like therapeutic windows and just not thinking about the sorts of questions that we would think about for pharmaceuticals. So this is part of the problem in this working in this area, and also there aren't many FDA-approved antioxidant medicines. There's a couple now, but there's also been a number of failures over the years, and we'll look at some of those in a minute. Okay, so let's just look at a few antioxidants that are likely contributing to suppression of oxidative stress in human tissues. And one we'll start off with are the carotenoids, best example of which is beta-carotene. Got this complex structure here with this long aliphatic chain with lots of double bonds. It's a very coloured molecule. That conjugation there means it's very coloured, and so it confers this strong orange colour on a whole variety of fruits and vegetables. So whenever you see a yellowy orange colour in a piece of fruit, you can often assume there's a carotenoid of some kind. There's hundreds of them. It's a very broad chemical class, and but they're pretty well everywhere. It's important in terms of beta-carotene, this structure here, as a precursor for vitamin A. So that's very important for good visual health, good intake of vitamin A. But it also has this additional ability to quench free radicals, and this is particularly relevant to superoxide and to a lesser degree probably peroxyl radicals, but it's able to delocalise electrons across that long conjugated system. So that's one of the reasons why it has antioxidant efficacy. Okay, but this is, again, so people thought, well, maybe we should be taking beta-carotene as a supplement. Those clinical trials have not gone well. It actually increases your risk in some regards. So we'll look at some reasons for that in a minute. Another well-studied antioxidant is alpha-tocopherol, which belongs to a class of tocopherols, and you can see them in terms of their chemical structures here: alpha, beta, gamma, delta, tocopherol, just depending on the positioning of the groups on the aromatic ring mainly, number of hydroxy groups and methyl substituents and the location of them. Alpha-tocopherol is probably the strongest antioxidant within the class. And we all get this product if we consume diets rich in vegetables, lots of it in leafy green vegetables like spinach and kale, but also in nuts, fruits, vegetables like avocado, olive oil as well, also rich in this source. So this is quite a strong scavenger of peroxyl radicals, and it's a classic chain-breaking antioxidant in that it traps peroxyl radicals and minimises their abilities to initiate lipid peroxidation. So that's the definition of a chain-breaking antioxidant. It blocks that initiation phase. And it's well suited to do this because this long hydrophobic tail on alpha-tocopherol anchors it in the membrane, and so it's able to trap peroxyl radicals within the membrane environment of cells. But it's also got this polar head due to the hydroxy group, and that confers the reactivity of alpha-tocopherol with free radicals, because remember the phenolic group is very important to trapping free radicals, because hydrogen undergoes abstraction. And so what happens when this molecule traps a peroxyl radical is we get another radical being formed, alpha-tocopheroxyl radical, where this oxygen here can be delocalised across this aromatic ring, and this is what makes it such an efficient antioxidant, because that radical is very unreactive. It doesn't attack lipids, so it's a true detoxification. And as with some other free radical scavengers in vivo, it can attack another peroxyl radical and undergo a further oxidation to form a quinone metabolite. So this para-quinone structure here is the ultimate product of trapping 2 electrons by tocopherol. And this can be found in urine. The quinone, it undergoes glucuronidation excreted in urine, so it's a potential biomarker. Not much of it there, but you can find it. Now we come to another low mass direct acting antioxidant that's very important historically, and that's ascorbic acid or vitamin C, and it's of course very historically significant. I'm sure you know the story about this. James Lind, famous Scottish naval surgeon, was one of the first to associate deficiency of ascorbate intake, vitamin C intake, in ill health, in sailors and maritime workers in days gone by. So in the age of sail, when European cultures started building ships and sailing out to discover the world, the sailors on some of those long distance voyages came down with a condition known as scurvy. It's a horrible condition. The hair goes all weird and sort of scraggly, and teeth start falling out, your gums become very spongy and bleeding from the gums, and as well as joint pain. So it's not a very pleasant condition, but Lind was one of the first to realise you could treat this by, if you took along lemons and limes in particular, and gave the sailors repeated consumption of these citrus fruits during the voyage. They were able to keep their teeth. So by the time like Captain Cook came and discovered Australia, they were well stocked with citrus fruits. It took a long time for that sort of recognition to be adopted, and there's sort of fascinating accounts of the suppression of that sort of knowledge by the naval leadership in Britain. It's quite an interesting story. Okay, so what's going on here? Well, ascorbate is not made in primates, and so we need a dietary source of this substance, and it gets accumulated via our GI tract via a sodium-dependent transporter, and it's actually saturable, so there's probably not a lot of point in consuming high doses of vitamin C, because it won't get out of your gut because this process is saturable. Okay, so why is it important as a radical trapper? Well, it's a bit like we saw with alpha tocopherol. Ascorbate can trap a couple of free radical species, in this case superoxide radicals, and convert them to hydrogen peroxide, which is then detoxicated by catalase, and so 2 radicals can be trapped by a given ascorbate molecule to form dehydroascorbate. The first step is the fastest one in chemical kinetics. Okay, now one of the interesting things that's been found quite early on in this study, in the study of the biological properties of vitamin C, was that in those, if you tried to reproduce that sort of scurvy condition in animals by starving them, so if you do this in, I think in guinea pigs, they can't make their own, but you can also induce it in rats to some degree by feeding them a ascorbate deficient diet. But one of the things people found was you get a vitamin E deficiency at the same time, so alpha tocopherol levels fall at the same time as restricting ascorbate intake. So what's going on here is that it was a discovery of the important cooperative role between these 2 small molecule antioxidants in protecting biological systems against oxidative stress. So what happens is that alpha tocopherol is particularly good at trapping greasy radicals, so those lipid peroxyl radicals, that long greasy fatty acid tail generated in membranes. So alpha tocopherol can detoxify them to form the lipid hydroperoxides, but then the tocopheroxyl radical formed during that reaction then gets reduced by ascorbic acid, and ascorbic acid is very water soluble, it's in the cytosol, so this reaction probably happens at the interface between membranes and cytosol. And then our old friend glutathione, which we encountered in first semester as a important trapper of electrophilic metabolites, it then acts sacrificially to regenerate ascorbic acid by reducing the ascorbyl radical to ascorbic acid. In the process, glutathione gets oxidised to form dimers, GSSG, and these eventually get exported from the cell. Okay, so this is a cooperative system that helps biological systems detoxify membrane phase the free radicals, and it's a way of protecting all those polyunsaturated fatty acids that we considered yesterday that are so vulnerable to lipid peroxidation. And polyunsaturated fatty acids are important because they confer a lot of fluidity on cell membranes. We need them because they have lower melting points, a lot of boiling points than saturated fatty acids. So they have an important role to play, but they are vulnerable to free radical attack, and this is a system that tends to protect them. Now, another class of antioxidants that's also acquired by our diet, like ascorbate, are the complex family of molecules known as polyphenolics and also flavonoids, an important class of them. And in a way, what we're doing in our labs this first half of semester in our drug discovery project is we're looking at some of the chemical analogues of benzoic acid, hydroxylated benzoic acid. And one of the molecules in your library is actually gallic acid, which is a naturally occurring substance present in green tea. But we're looking at trying to make it better as an antioxidant. Now, so we consume these every day, polyphenolic compounds, the whole structure of them, and like alpha tocopherol or vitamin E, they have hydroxy groups attached to a ring, but there's lots of them. So this makes them very efficient, highly potent inhibitors of lipid peroxidation. They're very good at quenching the peroxyl radicals within the membrane. Okay, so we probably ingest milligram quantities of these things if we drink lots of tea and fruit juices, and even red wine, white wine also contain different types of polyphenolics. And there's been a lot of research over the years trying to work out whether this sort of dietary source, particularly from red wine, is important in protecting against cardiovascular disease. There's been long-standing epidemiological observations that particularly males in France who eat a lot of fat, which you would normally expect to predispose you to cardiovascular disease, coronary heart disease, actually have a low rate of it. And the suggestion has been that it's because they drink a lot of red wine, which contains polyphenolics. Now, I'm not sure that theory is necessarily the only explanation for this so-called French paradox, but it might be a factor. Okay, so the reason is that these molecules are such good trappers of free radicals, and they form these radical species after reacting with an oxygen, reactive oxygen, to form these one electron reduced radical species that are able to delocalize that unpaired electron over that aromatic ring and that structure. So that's what makes them unreactive radicals, so they don't further react. So it's a true detoxification of a reactive oxygen species. Now, so when all these discoveries were made, people thought, oh great, we should, these free radical trappers are the bee's knees, best thing since sliced bread, maybe we should supplement our diet with them. But unfortunately, I think we began realising 10, 15 years ago that supplementing your intake with some of these small molecule antioxidants doesn't necessarily result in good outcomes, and may actually have bad outcomes. And so one of the first studies that showed this was in smokers, and people thought that maybe if you took beta-carotene and vitamin E, you could suppress some of the oxidative stress associated with tobacco smoke and might make health outcomes better for people. But unfortunately, some of that early epidemiology showed that people who supplemented with beta-carotene probably died at higher rates. And this was quite an unexpected finding. But we won't go through all this, but various large clinical trials, particularly in the form of meta-analyses, where people pull suitably appropriate clinical trials and epidemiological studies and look at large data sets to see what effects vitamin E or or beta-carotene supplementation has. In a lot of these studies you can see high-dose vitamin E might increase mortality, high-dose vitamin E may increase all-cause mortality, vitamin E does not affect cardiovascular disease-related outcomes, no effect. Beta-carotene, vitamins A and E may increase mortality. So more harm than good. These sorts of conclusions reach from doing these meta-analyses of vitamin supplementation clinical trials. So what's going on here? Quite a surprise to people because we were thinking that this might be a way to improve human health on a population scale. And also at the same time, based on that conviction, people were doing all sorts of work in laboratories, chemists, trying to make better antioxidants. So some of this work involved making better natural antioxidants. So when we realised that the mitochondrial respiratory chain is the main source of free radicals in aerobic cells, people came up with the idea, well, why don't we try to convert some of these antioxidants into mitochondrial penetrating chemical versions? And so you can find a lot of these still available. MitoQ, for example, where they put these triphenylphosphine tag on a molecule, these three rings here with a phosphine there. These get accumulated across the proton motive force of or across the gradient of the inner mitochondrial membrane, so it's a way to get a molecule into the mitochondria. And they've had some interesting results in animal studies, but they've never really gone anywhere, I think, as human treatments. Also, people have tried to make better catalase and SOD, superoxide dismutase mimics, by stripping these big proteins down just to the active site, which often contains an organometallic structure where the free radical trapping occurs. So people thought maybe we could come up with some orally active molecules, and they too haven't done particularly startling effects in terms of clinical development. We won't go through all of these, but certainly down the bottom here, modest clinical translation of a lot of these molecules that work quite well in vitro. But when you start testing them in animals or in humans, they've often had disappointing results. And also one of the problems in getting these molecules onto the market has been some very bad experiences. I'm trying to remember when this was. It was probably about the early 90s, I remember. This AstraZeneca molecule, Cerevive. And it's a peroxide radical trapper, a spin trap type molecule, coming back to the these spin traps here, able to selectively trap specific types of radicals. So that's where this molecule came from, a spin trap that, where am I going? AstraZeneca seemed to think it worked pretty well in stroke. So stroke involves lots of oxidative stress. What happens with a stroke, obviously, is you get a occlusion to a blood vessel, bringing blood to the brain, and you can break that blood clot down to treat the patient after a stroke. But when the oxygen rushes back in, oxygenated blood, you get this burst of free radical production and damage, and a lot of the long-term problems in people, the neurological deficits are associated with that burst of free radical damage. And a lot of acrolein and other lipid peroxidation products form also in that so-called reperfusion injury, it's called, when the oxygen-containing blood rushes back in. So you can reproduce stroke in animals, and AstraZeneca convinced themselves that this particular molecule was good for treating stroke in an animal model. But then when they got into phase 3 clinical trials, and that's where drug discovery gets really expensive because you've got to involve lots of hospitals and significant number of patients with a disease of interest, stroke in this case. But unfortunately, this drug performed really poorly. And when people went back and asked the questions, they realised that it wasn't well justified. This molecule should never have got to clinical trials. It didn't have good CNS access, so it didn't get into the brain. They also didn't incorporate ROS-related, reactive oxygen species-related biomarkers. We'll come back to that in a minute. So no one even knew if they were achieving antioxidant effects in the patients treated with this drug. And also people went back and looked at their animal models and they realised they are pretty poor models of human stroke. So several factors there contributed to the failure of these molecules, and that's why ever since, unfortunately, this has left a bad taste in the mouths of people in the pharmaceutical industry towards antioxidants, and so there's more scepticism about an antioxidant being useful as a clinical treatment. So this has led to what we call translational roadblock, whereby plenty of scientists have made promising molecules and even shown they work okay in animals, but there's been poor human translation. And I think Liz Johnson's going to talk to you about this pharmacology and translation issue. What does it take to get a molecule from a promising animal model into a human trial? Now, what are some of the problems then with translation of antioxidants? Why are they performing poorly in humans? And one of the reasons is just this problem, and I think I mentioned this in my very first lecture, that we didn't anticipate, is that reactive oxygen species aren't always bad guys. And this is this point I made, if you listen to my first lecture, I said I'm going to focus on the harm they do, but bear in mind they also have some positive roles to play. That is, reactive oxygen can play important signalling roles within tissues to keep some pretty important physiological processes ticking over. And so when you start swamping a human with high doses of antioxidants, you can suppress those beneficial roles, those beneficial physiological roles of low-level reactive oxygen species production. Okay, so for that reason, we started seeing side effects in people with antioxidant treatment that we didn't expect because of this problem, disrupting healthy roles. And one of the key roles of free radicals, normal physiological roles, is in the immune system. So if you shut down free radical production by the immune system, then you can leave the patient vulnerable to infections, unable to fight off infections. So that's one of the classic side effects of antioxidant intervention, increased risk of infections. And we now know this too, that some of those molecular participants in producing free radicals in cells, remember we talked about the NADPH oxidase system, very important in immune cells. And we know now that if you knock out some of these NOx proteins that produce superoxide in immune cells and other cell lines, you start seeing disruption of a variety of different physiological processes, including immune responses, angiogenesis, and responses to ischemia, and even sperm motility in males. So blocking in male infertility is a potential side effect of overdosing with antioxidants. Okay, so, and I just found this paper too the other day. It sort of confirms some of the clinical trial conclusions people have made about looking at high dose vitamin E. In this animal model, an ischemic stroke model, in animals, they found that they exacerbated post-stroke increase in markers of oxidative injury and neurodegeneration, the presence of elevated brain alpha-tocopherol, vitamin E, established that at supraphysiologic doses, that is high doses, alpha-tocopherol potentiates neuroinflammatory responses to acute ischemic stroke. So it's potentiating the role of inflammatory cell injury after stroke. So this is what people didn't appreciate in days gone by, how important free radical production could be in some cellular contexts. It's not all bad, so shutting it down sometimes results in negative outcomes. And then there's another issue that I mentioned earlier in the lecture, and that is that a lot of these clinical trials of antioxidants, people really haven't gone to the trouble of just asking basic pharmacokinetic questions. Does the dose of the antioxidant that you're administering, how does it behave in the body? What's its availability within the gut? What's its distribution profile? How quickly is it metabolised and excreted? And do you actually get a meaningful concentration, a minimal effective concentration being achieved within the target tissue over a sufficient period of time to get a suppression of pathology caused by oxidative stress? And are you avoiding doses that cause toxicity? So this is just basic stuff in clinical pharmacology, but when you go back and look at clinical trials of antioxidants, no one ever really addresses these sorts of basic issues. And then the other really important question is, are we actually incorporating clinically relevant appropriate biomarkers into a clinical trial to make sure we're giving the right dose of the antioxidant and that it really is producing an antioxidant effect that's relevant to the disease? Okay, so a biomarker, of course, is a molecule, some substance that can be measured in the body and predict the outcome or incidence of disease. And we're wanting clinically relevant biomarkers, ones that are specific to a particular disease, have prognostic value, that is, in the sense that if you can measure a certain level of a biomarker in a blood or urine, you can say that person is at risk of a certain severity of the disease. So these are very important. But when it comes to oxidative stress, this is a tough question, because remember, we've been emphasising during these last 3 or 4 lectures, free radicals damage lots of components of cells. They damage lipids, they damage proteins, they damage DNA, and within each of those categories they produce lots of different types of modifications. And so which one are you going to use as a biomarker in your human trial to make sure that your antioxidant really is suppressing those sorts of oxidative damage? So if you go back and look at the literature and have a look at the disease settings in which antioxidants are being tested, and you ask what were the biomarkers being measured in those clinical trials and were they appropriate, you can see highly patchy. So the black indicates yes, let's say in a CVD, cardiovascular disease trial, they did measure, some clinical trials have measured appropriate biomarkers, lipid-derived ones, for example, but then there's others that have been skipped. And you can see for a lot of these diseases, extremely selective selection of biomarkers and exclusion of a whole lot of them. Okay, so that's just a sad reality. We don't often know which ones we should measure. And so we don't really know whether the doses of antioxidant we're giving really do achieve a suppression of relevant biomarkers in that particular disease. And it really does matter. I just found this study of a Dutch study from a while back now, but they measured ascorbate levels, vitamin C levels, and clinical trials of vitamin C often haven't been very effective, and some of the biomarkers such as T-bars we're going to use in your lab today, they've used in clinical trials of ascorbate, and they never show much change. And so this makes it hard to know whether in a clinical trial people really are taking the appropriate dose of vitamin C regularly, and what whether they really are fully complying with study protocols. But in this Dutch study, they actually measured mercapturates. Remember that mercapturates are formed from aldehydes reacting with glutathione that then get processed in the kidney and form these N-acetylcysteine derivatives via the glutathione conjugate. And if you measure those in urine, then you do see this vitamin C related suppression of lipid peroxidation with this clinically relevant dosing of 500 milligrams twice daily. So if you measure the right endpoints, maybe you can see antioxidant outcomes with vitamin C. And so measuring these sorts of metabolites in urine might be a way of confirming that patients in a clinical trial really are taking the recommended doses of the vitamin C. Now also, there's been a bit of better news on the clinical front, and a drug that we're testing next week in our lab as a carbonyl trapper has been approved in the last decade, in the last few years in United States. It's been used for a while in Japan. But this is a darivone. So people claim that it's a reactive oxygen trapper. I actually think it's probably an acrolein trapper, and we'll be looking at that in our lab class next week. But it was approved 20 years ago in Japan for treating stroke, and it turns out Japanese researchers believe acrolein is a major mediator of stroke injury during ischemia and reperfusion injury, but so they use a darivone. It's interesting that it works pretty well in stroke. But then in the US, they've approved it in 2017 for treating Lou Gehrig's disease. This is motor neuron disease, the American name. And it's not that great a drug. It's got some issues with it. You have to take it via infusions, daily infusions that last for 60 minutes for 2 weeks, and then you have 2 weeks off and do it again. It's a pretty unpleasant dosing regimen, but some limited efficacy. And also, interesting results are being obtained with indirect antioxidants too, and these seem to be getting closer to the clinic, although there's plenty of hiccups. It includes things like sulforaphane, naturally occurring, and a synthetic molecule bardoxolone, and a few other drugs like DMF being approved too for treating neurological conditions. So what do these molecules do? Well, they essentially liberate NRF2. They dissociate, they modify KEAP1, which normally binds NRF2 and sends it off for proteolytic breakdown by proteasomes, but the binding of a molecule to the KEAP1 disrupts that process and liberates NRF2. It makes its way into the nucleus, and it activates the antioxidant response element and switches on hundreds of genes, antioxidant genes and phase 2 enzymes that metabolise antioxidants. Okay, so this is interesting because sulforaphane is derived from broccoli and broccoli sprouts, and so this is a naturally occurring substance that can also switch on this particular pathway. So these 4 NRF inducers are getting assessed in clinical trials in all sorts of different clinical contexts, and I just put this in because I thought this might be helpful to you for your deciding which disease of interest you want to follow up. It's a spectrum of conditions that have been associated with oxidative stress and are being studied in clinical trials. Okay, so sulforaphane, there's tons of it in broccoli sprouts, or Brussels sprouts rather, that people like to put on their sandwiches. This is a good way to get huge quantities of this substance in your diet, and there's less in a mature broccoli and also a bit in Brussels sprouts. Okay, so and then also this molecule that we thought for a while was getting closer to the clinic but has been going through all sorts of ups and downs is bardoxolone, and bardoxolone has a really tortuous clinical history. Back in 2012, Abbott purchased this molecule from a small startup company for a whopping amount of $400 million at the time. That was a lot of money. But it's been through a whole lot of clinical trials, some of which have broken people's hearts, despite showing hints of positive results. And but just about a year ago, a FDA rejection of use of this molecule in chronic kidney disease in people with a chronic kidney condition. So hopefully this molecule will eventually get there, but it's a strong NRF2 inducer. and the idea is that it'll be useful, particularly in people with clinical renal disease. Now, just to wrap up then, and I'm almost out of time, but people are now beginning to realise that if we're going to ever see antioxidants as widespread clinically used medicines, we've got to get a lot more intelligent, and just using an antioxidant to suppress all reactive oxygen species is not a good idea, because it has detrimental outcomes, because it screws up healthy roles or healthy functions of low levels of superoxide and other free radical mediators. So just giving massive doses of exogenous antioxidants every day of your life is probably not a very good idea. So what people are realising, we need to start working out where are the reactive oxygen species coming from in a particular disease, and can we come up with inhibitors that block that without interfering with healthy production of superoxide. So we're getting smarter, and people are realising now we need to target whatever deleterious process is producing reactive oxygen, rather than just shutting down all reactive oxygen production in vivo. And another concept too that's emerging is perhaps we should be restricting the use of direct antioxidants for conditions where there's an acute disease, where there's an acute overproduction of reactive oxygen, so that we don't see some of those harmful effects that occur when you treat people with high doses of antioxidant for a long period of time, chronic treatment. So perhaps we'll be able to reserve direct antioxidants for short-term diseases, acute conditions, and then maybe we should be using indirect antioxidants that have a bit of a milder action by boosting endogenous antioxidant enzymes via that NRF2 pathway. Perhaps they are the ones we should be using chronically over a long period of time to reduce the susceptibility to chronic disease. So that's just one of the paradigms people are working on at the moment. And then the other paradigm that we're particularly interested in with my collaborators in Matt Piggott in chemistry and some friends in the US, what about carbonyl trappers? Rather than targeting the reactive oxygen species, what about targeting the reactive aldehydes, the toxic aldehydes formed from lipid oxidation? So I'll talk about that in my lecture on Thursday, and we'll also explore that in next week's lab. Okay, thank you very much for your attention, and I'll see you later on in the labs this afternoon. Thanks.